Okay.
Yes, there you go. It's already on.
Okay.
All right.
So I think we can start up. So hello to everyone who joined us today for this seminar. We are glad to have you with us today, Professor Olivera. Thank you so much, Professor. So Professor Olivera have a PhD by the Stanford University and also was a postdoc fellow in the same university. And nowadays, Dr. Finn is a distinguished professor and chair of the Department of Immunology and the Department of Surgery at the University of Pittsburgh. So she was the president of the American Association of Immunologists from 2007 to 2008 and was awarded the AAI Lifetime Achievement Award in 2016 and was elected a distinguished fellow of AAE in 2019 as well. So in her lab, the long-standing interests center on identifying specific mechanisms of human anti-tumor immunity and cancer immunosurveillance. So they studied the immune repertoire in cancer patients and in healthy individuals at risk for cancer and the factors that influence that repertoire. And now this study developed by her group supported multiple clinical trials of a vaccine in patients with breast, colon, and pancreatic cancer. So Professor Finn, thank you so much for joining us today for the seminar. Just remember to our audience that will be open to questions after the presentation. So Professor Olivera, thank you again. I pass the floor to you now.
Thank you very much for that nice introduction. And I should just say that I welcome questions also during the presentation, because sometimes if you don't understand something, then you will not understand the next 3 slides. So it's better to stop me and ask a question. I really like interruptions, and because they direct me to where I should be more clear. So feel free. I talk to students all the time. It's my favorite thing to do. I've never said no to students, and I do like them to get engaged in a discussion with me. As I was saying in the beginning, I wish I were with you, but I at least can show you where I am now. I'm in my office in this building right here on the 10th floor. And this is the University of Pittsburgh. It's very close to the downtown of Pittsburgh. Downtown Pittsburgh is called the Golden Triangle. And it is a very geographically important part of the United States. It has 2 rivers, one coming from the south, one coming from the north. And this is where they form the River Ohio, then it goes into Mississippi, et cetera. So very important rivers of the United States. And this here, because it's the beginning of the River Ohio, is called the head of the Ohio. So as you can see, it's a very geographically not only important, but also very pretty place. And I hope some of you have had a chance to visit, and if not, will have a chance to visit. A few years ago, we had an AAI meeting here, so some of you may have made it. I have been in Brazil, and let's see how it's not okay. As you but not more than the capital. So not the capital. I'm sorry. During the Immunology Congress, which was now I think in 2007. So it's been a really long time. So I would have welcomed being with you. I think you are in Sao Paulo. Is this? Are you in both cities?
Both cities.
Okay.
Yeah, part of the audience is in Rio de Janeiro and part here in São Paulo.
Okay, very good. So Rio is the only place that I know. And when I was there in 2007, I was sure I was going to come back very soon and see more of the country. And of course, I didn't. But anyway, there's still time, right? So I'm going to divide. I gave you a title that really belongs to only the part 2 of the talk. But then when I was putting the talk together, I thought I would give you a little bit of a flavor of some of the more recent work that we've been doing. And so I've divided the talk into 2 parts. Something about early detection according to the immune system, because people talk a lot about early detection so that we can intervene very early in cancer development. And so I want to show you what we think in terms of early detection. And then I will change to the main topic of the talk, which is the cancer vaccines that we are now developing for a prevention of cancer, be it post-early detection to prevent progression, so that's called interception, or to prevent it to begin with. So the first part, or actually just sort of to introduce the whole talk, you all know this particular graph, this particular picture. We've seen it so many times. It has to do with our idea now of immune surveillance of cancer and the various outcomes of that immune surveillance. In other words, there is no doubt anymore that the immune system has a lot to do with cancer, and that the immune system's one very important function is to keep transformed cells from becoming tumors that threaten the integrity of the organism. However, everything that we know really is based on this last part, which is a disease that has already escaped the immune surveillance. And there we have all the members of the immune system present. The problem is there is such a very highly immunosuppressive environment that has finally evaded this immune surveillance that has been in place from the first changes from normal to abnormal that have led either to elimination, where again, all the immune system members get together to get rid of these very early 2B tumors, or if they can't get rid of them, they keep them in equilibrium. And you know that sometimes by the time the tumor is diagnosed, there's a thought that it's probably been there 10 years, 9 years, 5 years, etc. All that depends on how well the immune system was able to keep it in this equilibrium. But again, as I said, we mostly look at people who are diagnosed with cancer, and everything that we have learned about the immune system in this particular stage is that it is highly immunosuppressed. And while there are specific T cells and specific B cells and antibodies, etc., there are other participants that, in this chronic inflammation, chronic immunosuppression that keep the adaptive immunity from being effective and from getting rid of the cancer. Nevertheless, what we have also learned is that even when this tumor is removed, the primary tumor, the fact that there were tumor-specific responses present and not working at the time, they still make a big difference in terms of the rate of recurrence, overall survival, etc. So the immune system, when helped, can really do its job. And of course, as you know, checkpoint inhibitors are the one way that we help the immune system sort of reset, get back to being able to see the tumor cells and get rid of them. But this is a part that we really don't know much about. So we say there's normal tissue, and all of a sudden there's transformed cells, and then something happens. But in between here, there is a lot of possibility to understand what happens between here and here and to intervene. So, because even here, by the time you have a lot of transformed cells, it may be very difficult for the immune system to intervene, and this elimination may not be a default reaction, and mostly equilibrium may be what's happening. So we ask the question, when is it that the immune system sees something happening that is not yet a cancer, but it could become cancer? When does it become obvious to the immune system? And so, as you know, everything really starts with innate immunity. So some macrophage or dendritic cell needs to see something wrong and to present it to T cells and B cells, etc. So we decided to look at macrophages. And you know that in the escape phase of tumor, when the cancer is still already there, we think macrophages are bad. And this is what this cartoon actually says. Didn't I see you associating with some tumors the other night? He said, who, me? He said, tumor-associated macrophages are the worst. And so we know that macrophages can induce tremendous immunosuppression in the tumor microenvironment, and actually with their products help the tumor progress and grow. But the macrophages are not TAMs, tumor-associated macrophages, in the beginning, when the tumor is just beginning to grow, or something that will become a tumor is beginning to happen. So, as I said, we asked this question, when do macrophages see that a cell is not normal anymore, and then they should alert either get rid of it itself or alert the adaptive immunity to come and clear the site. And we were very much inspired to do this by a colleague of mine, Rachel Gottschalk, who ended up being a collaborator on this project, because she had a few years earlier asked that same question in terms of infectious disease. Because it's interesting, the macrophages actually see and they get triggered through their toll-like receptor 4 by bacteria, by infectious agents. But they have two ways of reacting. One is that they go into an inflammatory gene expression program. And the other one is basically that they ignore it. So Rachel asked the question, what is the difference? And what happened to be the difference here is the amount of the product, of the amount of the ligand that is seen by the Toll-4 receptor. So as long as there is very little, macrophages ignore it, but there is this threshold where then they go into full activity. And then they make TNF-alpha, they make IL-6, they actually activate, and they can destroy they can eat the bacterium, or they can, as I said, signal and actually they do both. So we said, if that is the case, and actually I very much like this idea because I always think of the immune system not only as qualitative, that something's foreign versus self, etc., or dangerous, but as quantitative as well. So maybe we can ignore things that don't reach a threshold of danger. But then when they reach a threshold, we really have to respond. So I thought, if this is happening with bacteria, what can we actually find that in the case of cancer? And we had a nice model, in vitro model, that we thought we would use a system. And these are 4 breast cancer cell lines, actually 4 epithelial breast cell lines. This one is normal, and they're all derived from the same individual, so they're all on the same genetic background. And however, this grows in vitro, but doesn't form tumors. In mice, doesn't form tumors in soft agar, so it's actually immortalized, but fairly normal. Then there is a hyperplastic state where it forms these sort of premalignant knobs, but not tumors. Then there is the next stage of pre-malignant development, which in breast cancer, it's like the ductal carcinoma in situ, which is not cancer, but it is a pre-malignant lesion. And then there's a cell line that actually copies very well the invasive cancer. So we thought, okay, what if we purify some peripheral blood monocytes, generate macrophages, so fresh macrophages, or we actually had also a cell line that it's a monocytic cell line that we can treat to make into fully developed macrophages. So what if we just incubate them with these cell lines and see what they do? And what we found was really interesting, and that was that they ignored the immortalized one. They ignored the first premalignant hyperplastic cell line, but then they actually completely changed what they did when they detected something that looked like DCIS, and then of course they continued to react against cancer. So both primary macrophages, and meanwhile, and we are measuring TNF-alpha and we are measuring IL-1 beta, very pro-inflammatory cytokines, while actually IL-10 that keeps the macrophage quiescent and the environment quiescent is doing just the opposite. These are cell lines that keep them quiescent, but this is where IL-10 stops being made and instead pro-inflammatory cytokines are made. So that was interesting and it kind of was pleasing as an idea because, again, not every change in a cell from normal to abnormal is worth getting the immune system all excited. So this threshold was interesting also because we looked at doublets to see when do macrophages actually bind. And what we saw, and all of these say the same thing, is that they bind, they make conjugates with a normal cell, with a hyperplastic cell, but that's all they do. They make conjugates. But look what happens just to the next step. Not only do they make conjugates, but they swallow the cell up. And then, of course, they phagocytize the tumors as well. And you can see, again, this threshold from hyperplastic to pre-malignant is the threshold that the macrophages respect. And here is, we grew the cells in 3D, in spheroids. And you can see again, the green are macrophages. These are tumor spheroids, or actually these are the normal cell, epithelial cell spheroids. And you can see that macrophages are just sort of looking at it and not getting excited. You can see the difference in what these spheroids look like. This is the hyperplastic state. Again, the macrophages are not that excited, but look what happens here and here. Here they invade. They're still sort of, not exactly doing anything very bad, but the cancer, they are destroying it. And so this is just quantifying all that. So again, this is the threshold between just hyperplastic, but not abnormal, and then the first stage of the pre-malignant disease. So we wanted to know what was it that was expressed on the pre-malignant but not hyperplastic cell that would excite these macrophages, or what was it that the cells were making? So these are the below threshold cell lines, these are the above threshold cell lines. We did a unbiased differentially expressed genes analysis, and we found multiple really interesting genes, but only this small group of genes was really respected sort of our threshold. So these are the genes that were expressed in the pre-malignant and the malignant cell, but not to that extent in the previous stages of transformation. So we were not really, it didn't give us as much. So we thought, well, maybe it doesn't always have to be differential gene expression. It can also be different protein processing, amounts of protein, right? As we saw in the TLR4 story. So we actually analyzed every cell against the normal cell, and through these 2D gels, and this is just an example of these gels where you resolve protein. So these are total proteomes of each one of these cell lines compared to the normal cell line, and you resolve all the proteins by molecular weight and by isoelectric point. And you can see here in red where all these, so green were the same, yellow were overlapping, and in red were the new proteins that happen at the threshold. And those were tubulin beta, gelsolin, serpin B1, annexin A3. And you can see that in each case, they are expressed in the T1 and CA1, the pre-malignant and the malignant cell line. So then they happen to be cell surface proteins, a lot of them. And you will see here that the 10A, the normal one, and the neo-T, that's hyperplastic, are all sort of in background expression of these molecules. But here's the threshold and here's the tumor. So here's the pre-malignant and here's the tumor, pre-malignant, tumor, and you can actually see the difference. And here they are statistically significant. So we were able to get antibodies to two of these molecules that are commercially available, and we pre-treated, actually exposed the macrophages to the various cell lines in the presence of these antibodies, and you can see that that in fact for these two CEA and XNA1, the antibodies can actually block TNF alpha production by macrophages. And interestingly, and I'm just showing you bits and pieces, the paper is published, by the way, recently, I think sometime last fall. And but we actually looked at human tissue samples from normal breast, hyperplastic breast, and then DCIS, again, this is this threshold, and then invasive ductal carcinoma. And you can see that in terms of macrophage presence, so we looked at the presence of macrophages. Do macrophages infiltrate like we looked at the spheroids in vitro, right? Do they make TNF alpha? Do they express, do the breast tumors express SERPINB1, annexin A, and CEA, which were the molecules we identified as being interested to the macrophages. So again, you can see that in terms of macrophage infiltration, normal and hyperplastic are macrophage free. DCIS is very heavily macrophage infiltrated, as is in invasive ductal carcinoma. Where there are macrophages, there's TNF alpha production. Here we didn't see them very well. We have a little bit of TNF alpha production. Here we have a lot. And you can see it's by tumor and the macrophages, and we have a lot of macrophage TNF alpha production here. SERPINB1, you can see no expression here, no expression here, but a lot of expression here. And this is sort of positive, but not as much as this at the threshold. Annexin A1, normal, is fairly negative, very patchy in the hyperplasia, very high level of expression, DCIS and in IDC, and CEA exactly the same. So we are seeing that human breast cancer development goes through these stages that are recapitulated by the cell lines that we had, and in both the cell lines and the tumor tissues that we have, in normal tissues we have from breast cancer patients, we see this threshold of macrophage infiltration, activation, inflammatory cytokine production, etc. So just to conclude this first part, it is, the immune system is watching for this threshold between what's normal and what is becoming to be abnormal. And the macrophages, and I should say that I didn't show you, but dendritic cells are equally sensitive to this threshold, and we are doing more with them now. I think we have it in the supplemental data in the published paper. But the innate system that we are measuring by looking at macrophages appear to be tolerant to normal breast epithelia, as they should be, mostly absent from normal epithelia. But then there's a switch-like response to a pro-inflammatory state that happens between this benign hyperplastic and then atypical hyperplastic, this premalignant state, and then it's maintained in invasive cancer. Now, as you can imagine, at this atypical hyperplastic state, there's still, I think, the role of the macrophages is to call the adaptive immune system in. If it doesn't come, then these macrophages continue to create this inflammatory state and eventually become tumor-associated immunosuppressive macrophages. The same threshold applies to tissues, infiltration, phagocytosis, which I showed you we saw in breast tissue section. And then as we identified a few candidate molecules, there are many more that we are trying to work out right now. And I think they're danger signals. These are interesting because some of them, like you saw CEA, et cetera, are already identified as tumor-associated antigens by finding tumor-specific T cells, tumor-specific antibodies. And I think they do come because the macrophages have now presented these new molecules that are overexpressed to the T cells and to the B cells, and are trying to generate an immune response. So, and these same molecules I showed you are expressed on breast tissues. All right, so maybe I should pause here and get a few questions. I only see my screen, so I don't see if anybody has a question. You have to monitor for me.
Yeah, normally the students are shy, so we wait until the end. But okay. I have one question.
Yes.
So what about signals from cells that would be dying from apoptosis, like externalization of phosphatidylserine that wouldn't appear, I think, in your screen? Would you consider that this could modulate also macrophages in that tissue microenvironment?
Yes, phosphatidylserine was interesting to me. We didn't find it as a candidate. And part of it may be because we're looking at live cells. So you're absolutely right. This is something that phosphatidylserine is interesting to the innate system for sure. And also for viral infected cells, when they sort of turn their membrane inside out, they are immunostimulatory. And you're right, we should take a look at that. But just regular apoptosis, if it's not inflammatory apoptosis, is not triggering them.
Right. And another thing is that, of course, you look particularly to the breast cancer environment, but do you believe this annexin A1, for instance, would be a marker in all tissue?
I think certainly epithelial, right?
Right.
Certainly epithelial tumors and epithelial premalignancies. I think so. And it's actually been reported. A lot of these molecules have been reported in tumors, right? As sort of overexpressed, abnormally expressed, et cetera. So in the discussion in our paper, we sort of brought up everything that's known about these molecules, all from cancers. And they're not all breast cancer, absolutely not. They're all very different, multiple different, at least epithelial tumors. So we don't know whether, for example, melanoma or lymphoid tumors, right, you might find some other triggers, right?
Yeah, that's great. It's a great idea, great project. Thank you.
Thank you for asking. All right, so
sorry. I think Miriam has a question as well.
Okay, yes.
Yes, quick question. I wonder, because I was thinking about tumor metabolism that might change as well, and
yes,
I was wondering if you tried pre-treating or putting lactate or some other factors together with the macrophages and seeing if you change the threshold.
So we haven't. We did two things to change the threshold. One was to pre-expose, let's say, trigger the macrophages, LPS, activate the macrophages. And then, so that would sort of change their metabolism, and then expose them. What we got was exactly the same threshold, but higher activation level. Okay, more TNF, more, because they were primed, but the threshold was exactly the same. And the other thing that we did was we treated all the cell lines with neuraminidase, just like, because sialic acid is fairly inhibitory to some of the macrophage ligands, et cetera. We treat with sialic acid to see if we can expose further ligands, and maybe it's just that, right, from normal to abnormal, and nothing changed. So we did those two things.
Okay, yeah, because maybe depending on the size of the tumor, you might have. Lactate is something that I think that
absolutely
into the concentration and might move a little bit the curve.
Yes, and then I think this happens, very early, when I don't think we can really think about the size of the tumor. I really think this happens, fairly early when you're dealing with, clusters of cells that have changed. Yeah.
I see. Thank
but
you.
It's only a guess, right? We can only imagine. So, all right. So, the main topic, the cancer vaccines, and I said preventative, but it's really for cancer interception, for prevention, for risk reduction, and this and I wanted to specify that we're not talking about HPV, for example, or HBV, or the virally caused cancers are a wonderful target for preventative vaccines.
If I could just, I'm sorry to interrupt you, is that one of the students just posted a question in the chat.
In the chat?
Would you mind about the first part? Would you mind if we ask, or would you rather leave it for the end?
No, go ahead and read it to me.
Okay, she's thanking you for this exciting initial part. Her question is about the DC signaling. Do you see the same response for DCs, or if there was a difference in the quantifying the signaling?
Yeah. So to the extent that at the basis of our sensitivity, which is TNF alpha production, we did not see a difference. But we didn't really look at. So we now know sort of some of the molecules. We really don't know the ligands, and we have not looked at inside the macrophage what happens and inside the DC what happens. I think it'd be very nice to do that. We kind of imagine that that we know what happens to get TNF alpha production, right? So the closest approximation that we got to signaling, we didn't see a difference. But that is not to say that there wouldn't be. For example, you may know that the macrophages will eat a bacterium and kill it. Then the dendritic cells wouldn't. They just eat it, and then they present some antigen, but they don't kill it. So they do act very differently, and I think that those would be interesting things to look at.
Okay, thank you.
Sure. So you all know, if you are thinking about doing a vaccine, you have to know what the antigen is going to be. And when I got into this, which I did like 35 years ago, there was this thought that there's nothing on tumor cells that would trigger the immune system because the tumor is self, right? And the whole idea of the immune system was that the immune system is a self versus non-self discrimination. Then we started to understand that in fact, that is not, that's what transplantation is based on, but organ transplants. But what the immune system really wants to know is it dangerous? Is it going to hurt me? And so we had sort of a renewed interest in looking for what is it on tumor cells that the immune system sees as dangerous, and at the same time different and foreign. And when I got into it, the oncogene field had already understood that sort of the genetic basis of some of the tumors, so the KRAS, the mutated p53, the fusions, BCR, such as BCR-ABL that generate leukemias. And most currently, people are very interested in random mutations, all of which happen in the tumor and not in the normal tissue. The tumor is and often these are driver mutations, driver oncogenes, and their products should be perfectly good antigens. And that was the first, so that was the first idea. Let's find out, if what products does the immune system see from the oncogenes, and then we can generate the vaccine, immunize people against these oncogene products, and every time this happens, the tumor will be rejected, or the beginning tumor will be rejected. The problem was that we looked very hard with both. We could immunize animals to these molecules, but we could never get tumor rejection responses. We could those predicted molecules. We could. But in patients, in sera of patients, in T cells, in TILs, we could find maybe one or two, but we couldn't find enough cells, T cells or enough antibodies that are specific to these products. So there was something that these were potential antigens, but not really good antigens to target. And we now know it's because they're not really expressed at very high levels. The mutation is enough to drive the tumor, but not to generate a very good immune response. And as I just showed you with the macrophages, it requires a threshold of expression for the immune response to kick in. So they were all below threshold kind of changes. By far the most important antigens to the immune system we found were those that were overexpressed compared to normal cells. So no mutations, but high levels of expression compared to normal cells. And then you get a large representation of peptides from these molecules in class 1 and class 2 molecules of tumor cells, and you can activate the immune system. And if they're cell surface molecules, you activate macrophages, et cetera. And in addition, and sometimes just because there's this overexpression and uncontrolled expression, you also get different post-translational modifications. So even if it's the same amount, or mostly actually it's in overexpressed, you get overexpressed, overrepresentation of peptides, but also they look different than the proteins from, peptides from the normal proteins. So, and some of them, WT1, P53, HER2/neu is all based on overexpression, CEA, all on overexpression, prostate-specific antigen, the same thing. And we discovered mucin-1, MUC1, which I will tell you a lot more about in the continuation. And the other one was cyclin B1, both by virtue, cyclin B1 especially by virtue of overexpression, MUC1 an antigen by virtue of overexpression, as well as different post-translational modification. So it's a hypoglycosylated MUC1 that is a tumor antigen. And then there are others that you may know. The phosphopeptides can be different. You get different signaling, so you phosphorylate proteins differently, and then you get different phosphopeptides as antigens, citrullinated peptides, etc. So by 2010, 12, etc., we had lists of antigens that have been defined as tumor-specific antigens or tumor-associated antigens, and being able to be seen by the immune system, by both T cells and B cells. So, but here's MUC1, and I will give you a lot of examples that all based on our work with MUC1. We published this molecule as a tumor antigen in 1989. And have been working with it since then. And the reason it's a tumor antigen is that on normal epithelial cells, on all epithelial cells, it is expressed as this kind of molecule with a transmembrane, with a short intracytoplasmic tail, and this very long, tandemly repeated 20 amino acid sequence. It can be up to 200 or more tandem repeats, identical. And each tandem repeat has 5 glycosylation sites, serines and threonines, for all-in glycosylation. And in normal cells, this is expressed at low levels, but fully glycosylated. So this part of the molecule, this protein sequence, is never processed and presented to the immune system. It's completely hidden from the immune system because it cannot be processed by dendritic cells and presented. And we've published that already. In tumors, you get 400-fold or more production of this molecule. And it is very, very hypoglycosylated. So very few of these sites are taken up by sugars. And when they are, the sugars are short, truncated, and capped immediately by sialic acid. And so you have a lot of protein that can be exposed to antibodies. And this can be processed by dendritic cells and presented as both peptides, naked peptides, and glycopeptides. And the T cells see both the peptides and the glycopeptides. And antibodies are also found in patients. We generate them in mice, et cetera, against the naked peptides, as well as these tumor-specific glycopeptides. So we did, I told you we published this in 1989, and already in 1993, with my colleague, Mike Lotz, who is a surgical oncologist, and I just moved to the University of Pittsburgh, we put together a clinical trial to use this hypoglycosylated, actually just a non-glycosylated peptide of 100 amino acids, which is the 5 tandem repeats in a row, synthesized as 100 amino acids. Actually, this one is 105 amino acids, no sugars. And then we mixed it with an adjuvant that I thought was the best, which was BCG. You all know that's a fantastic stimulator of that Th1 type immunity in people. And we started a first in the world trial of a cancer vaccine based on the sequence that comes from you and is not mutated. So it was a lot of trouble trying to get this started in terms of the FDA approvals, and they were all worried. So we had to limit our trial to stage 4 cancer patients who have failed all other therapies. So this was not supposed to work. It was just supposed to be first in human testing. And we did it in colon, breast, and pancreatic cancer patients, a late stage disease. And we found some signs of immunogenicity of the vaccine, even in these very late stage patients, but we couldn't look at outcome or anything like that. However, it opened the door to many other future trials. And over the next, so this was the 1993 trial, through 2010, excuse me, we ran various trials. And you can see here, the MUC1 is an antigen on all epithelial tumors. So we could do pancreatic cancer, breast cancer, colon cancer, etc., prostate cancer. And so we did all this. In each case, it was 100 ml, but we changed the adjuvants as we even loaded it onto dendritic cells. We used a TOL3 adjuvant. You can see another DC trial. In each case, we could show some immunogenicity, very low level antibodies, very low frequency of new T cells, but very seldom was that correlated to outcome, disease outcome. So by 2010, I had had enough, and especially because when we started our first trial in 1993, and this is me in 1993, you can see how long ago that was, we said that we're testing this vaccine that can trigger immunity to 3 forms of cancer so that we can eventually prevent the disease. So Mike said that we hope someday to inject people at risk for developing cancer as a preventative. Finally, in 2010, I got to that point. We had done so much showing at least that it was safe, and so we were allowed to think differently with this molecule. Because we had in the meantime done a lot of mouse studies and monkey and rhesus macaques and even chimpanzee studies to show that the inducing an immune response to, these were all in healthy animals, so we can induce very good immunity. Inducing an immune response to this form of MUC1 does not attack normal MUC1, which hides that particular part of the molecule. So we decided to change how we do everything. So we said, we're going to take a new approach that will be preventative rather than therapeutic. So we will, and so rather than vaccinating, people would say, what are you going to vaccinate, 25,000 women, young women, and wait for 25 years to see if they develop cancer? I said, no, we're going to actually look at the pre-malignant setting and see if we can, keep it, enhance the equilibrium, keep the things from progressing to cancer. So we would target tumor antigens that are expressed on pre-malignant lesions, and MUC1 is expressed not only on full-blown epithelial tumors, but also on their pre-malignant lesions. And the hypothesis was that it should work much better because therapeutic vaccines are given in the setting of high immunosuppression in the tumor microenvironment, whereas here, there is no tumor microenvironment, and therefore it should be the vaccine should have a much better chance of working. So we started with one pre-malignant lesion that was beyond the threshold that I showed you for the macrophages. This is the last step before colon cancer development, and it is advanced colonic adenoma. And but the important thing about this is that it isn't cancer. When it's removed, when it's found on colonoscopy and removed, 50% of these people recur within 1 to 3 years. So you can look at the immunogenicity of your vaccine, and you can look at recurrence rate as your efficacy. So with a relatively small number of people and in a relatively short amount of time, 3 to 5 years, you can actually test your vaccine for both immunogenicity, toxicity, etc., and efficacy. So we went with that, and first we did and then the whole thinking was, let's go with MUC1, abnormal MUC1, because we know that the normal epithelium shows only normal MUC1. The small adenoma shows a little bit of abnormal MUC1. Large adenoma shows more, and then carcinoma and metastasis express very high levels. So we used to vaccinate here, which is sort of, the battle is already lost. So all we wanted to do is move the vaccine here, remove the large adenoma, vaccinate, see if any of this happens again, rather than, progressing. So we started this feasibility study in 2009. And with the patients, like I just described, we used this vaccine, and we used the TLR3 agonist, which works very well in our animal models. And all we wanted to know is safety, because you remove the adenoma and the people are now completely healthy. So you have to, they're not treated with anything else. So you have to show safety and MUC1 specific immunity. So we published this paper. If you want the details, you can look at that. But basically, we had a lot of responders, but really just 47%, which we showed that those who did not respond, I don't have the time to show you that data, had actually already increased level pre-vaccination of immunosuppressive myeloid suppressor cells. So these large adenomas are already setting up in half of the patients an immunosuppressive tumor microenvironment, telling us that we should go really even earlier in the colon polyp to colon cancer stage. But importantly, there was absolutely no autoimmunity, no toxicity, et cetera. And I can just show you what happens. So we had responders and we had non-responders. And you see this dotted line. This dotted line is the height of the immune response that we could achieve in cancer patients. And you can see how many fold, 35-fold in our responders when they're healthy, when there's no cancer on board. Of course, when there's immunosuppression, they're not responding. And in terms of the injections, the vaccine was given at week 0, week 2, and that's when everybody responded. Some people required 3 vaccinations to respond. This is a fairly aged patient population. And then the immune response goes down when there's no more antigen. And 1 year we boost and we generate an amnestic memory response. So we had immunogenicity, we had safety, we had memory, which is something that you can't achieve in the setting of cancer. There's no memory, there's no, and the immunogenicity is very low. So we went into then because of that, we were allowed to go into randomized, placebo-controlled, double-blind trial of exactly the same vaccine, the same setting, except now we had a placebo control. And we can then look at immunogenicity and safety compared to the placebos, but also at efficacy. And we powered our study to see 25% reduction in polyp recurrence by year 3. And then we did a lot of other secondary endpoints. But you can see that it took just 130 registered people. So it isn't a trial of 25,000, a prevention trial. And we opened this in 2014. We finished it by 2016. And I said it was a 5-year trial. We just submitted a paper to gastroenterology. And we repeated completely our study. So the red ones are placebo, the blue ones are the non-responders. So we had responders and non-responders. We also had MDSCs for driving the response or not response. But the important thing here is that the placebo controls, 66% had new polyps within 5 years. In vaccine non-responders, 64%, but vaccine responders, only 27.3% recurred with new polyps. So we had powered the study to see 25% reduction. We saw 38% reduction in polyp recurrence. So seeing these data, we, because as we were accumulating data during the trial, we also opened the trial now with no premalignant lesions. These are just smokers, current and former smokers who are very high risk for lung cancer. And so the study just finished and we're analyzing the data. But why we chose that is because the smokers' lungs, if you look at moderate dysplasias, are a very high level expression of abnormal MUC1. So we thought that if we can vaccinate smokers, that these lesions perhaps will be controlled and kept in their equilibrium and would reduce their risk for cancer development. I can give you a little preview of our data, and that is that current smokers, as well as former smokers who have stopped smoking within 1 to 5 years from our study, are very highly inflamed inside their lungs. A lot of them have COPD, and we were unable to get the kind of immune response from them that we have gotten from the polyp patients that just had polyps removed. So they're very highly immunosuppressed. And I just saw a study published that even to a COVID vaccine, smokers and former smokers responded much less than non-smokers. So this chronic inflammation, whatever it is due to, and in this case is due to smoking, is a very bad environment for the immune system and for generating new immune responses. Okay, so I'm getting to the end. So what we now know from these trials is that the MUC1 vaccine that is only weakly immunogenic in late stage cancer was highly immunogenic, safe, and what I just show you, effective in the setting of premalignant disease. We generate immune memory. And we also discovered that even in the premalignant lesion, there is immunosuppression, and we should be aware of that. When we do also anything, that people are talking now about doing actually checkpoint inhibitors in the premalignant setting, again, the MDSCs are not going to help them very much. So this has to be taken into consideration. I just to show you the potential of what we can do just with the MUC1 vaccine, the one that we have now. We can try to treat precursors to pancreatic cancer, to breast cancer, to multiple myeloma. This MUC1 positive MGUS is a precursor to multiple myeloma. These are circulating clones of B cells, and we can, get rid of them if we generate an immune response. Barrett's esophagus, so just with MUC1. But this is a long list of many premalignant lesions that we can diagnose very easily by imaging, by blood tests, etc. And they all have antigens that they express. And that, this has been now published to be, the HPV vaccines have been published now to be fairly effective in cervical lesions that are not cancer yet, CIN1, 2, and 3. So we have an opportunity to stop the cancer from developing, and we have the antigens that we've used not successfully in cancer, but could use them very successfully in premalignant lesions. And that is the goal that, that I'm following and hoping to open doors so other people can go in that direction. I can skip that and acknowledge my lab. This is their current version, but they change all the time, right? But people whose data I showed specifically are listed here in red, and these are my trial collaborators. And we do our trial to the National Cancer Institute Division of Cancer Prevention, their cancer prevention network, and that has been very good for our work. So I'm going to get out of here so I can see you all and you can ask me questions. Let's see. Stop sharing. All right. Thank you.
Excellent.
Thank you so much, Olivera.
And
so
if I can ask
open to questions.
Yes, if I can ask whoever can turn on the camera.
Want to raise your hands or send in the chat?
Whoever can turn the camera, I would love to see. Oh, Carla, I know you. Hi, how are you?
Hi, Professor. It's nice to see you again.
Nice to see you too. Carla was taking a course and I was her mentor for IUIS. Very nice to see you.
I would like to ask you a question, Professor.
Sure.
So, do you think that these early vaccines could work also on people with family history of cancer?
Yes. So I'll give you a concrete example. The Lynch syndrome is so that's really genetic risk. It's not so much family history, but from very early on, right? So you can test for it. So the trial. One of the colleagues of mine from Kansas City, who was the polyp trial, that was a placebo-controlled trial. It was also a multi-center trial. And so he was one of the centers. He then used our vaccine and proposed to do Lynch. So he's vaccinating people with Lynch syndrome to reduce, and very early on, to reduce the number of polyps that could then become malignant. And I would say, if we show efficacy and safety, then, you don't need to wait to get a, polyp by colonoscopy, et cetera. You can, if you have a familial risk, I have, and my father died of colon cancer, as in my grandfather, it would be nice to, and I'm 72, so I'm already immunocompromised, right? But it would have been nice at 50 to have gotten the vaccine, or 40 or something like that.
I see. Thank you. I have another question also. Do you think that we can associate something to the vaccine to maybe get those non-responders to respond to the vaccine? Yes. There is something.
Yes. Yes. So we are trying to think in terms of. So there are a lot of companies that are working on finding ways to get rid of myeloid-derived suppressor cells. And we don't have yet, because they're not that they're specific by their function rather than any target molecules that we have been able to identify. But we can maybe, overcome their inhibitory environment. So, for example, things like, and sort of metabolically, so things like arginine is very important for T cell function. They make a lot of arginase and destroy arginine. And glutamine is very important. Glutamase also. So what we're hoping to do is take our non-responders, put them on 2 or 3 weeks prior to vaccination on very high levels of arginine, which people take anyway sometimes, glutamine, things like that, to bring up, and then throughout the vaccination, and then again 2 weeks before the booster, and something like that. So that's what we are thinking of now, because everything that you want to do has to be in prevention, has to be simple and safe and cheap, because, I mean, for something like Lynch, checkpoint inhibitors are okay, but for just the regular risk, I think you don't want to go on checkpoint inhibitors, right? So, yeah, so we are very much watching what other people are finding, or discovering, and if we can put it into our trials.
I see. Thank you, Professor. It's nice to see you again.
Nice to see you too, Carla. One problem with clinical trials is that it takes a long time to start them, from the time you propose them. And by the time you start them, if something new has been discovered, you can't use it. Even, you have to do it the way you said you were going to do it. So that's always, hopefully, now things are happening a little faster. So maybe we'll do okay. Okay, there's another in chat from Bruno. Bruno, can you make yourself visible and ask me that question?
Well, just a second.
Okay. It's my one way to see you guys.
I can actually make myself available to ask the question, but I cannot open my camera because
Okay.
I'm recovering from COVID. I'm not in any shape to appear on camera. I'm sorry.
Got it.
But it was a really nice talk.
Thank you.
Very, very, very interesting. But one thing that's it's always very hard that you mentioned that before. With the clinical trials, you cannot test every possibility, and you cannot change course midway.
Yeah.
But with the results with the polyps, it's very interesting that you actually reduce the recurrence of adenoma. But in the placebo-treated group, you still have about 1/3 of the patients that do not develop adenoma.
Right.
So how unlikely is that the patients have these different immunological makeup, and that's what is conferring the resistance to polyps, and what the vaccine is doing is actually helping you identify these patients that have these somehow natural resistance
Yes.
to them.
Yeah, absolutely. You're right on the mark. I don't think that our preventative vaccine is priming, it's boosting pre-existing immunity. So I think that the people in the placebo group that have not recurred have pre-existing immunity. And it doesn't necessarily have to be to MUC1, but it can be to many other molecules that are normally expressed. And the other seminar that I could have given you has to do with the new new way we're thinking about immunosurveillance. And that is, it's immunosurveillance not only of cancer, but of health in general. And that is that there is a set of molecules, and MUC1 is a prototype of these molecules, that we have discovered as tumor-associated antigens, but you see that they are actually abnormally expressed in other inflammatory, febrile infections, viral infections. And if that's early in life, they generate a very good immune memory for these molecules. And then when they re-expressed on tumors, then you actually have their first responders. And so we have shown that in many, and if you're interested in, just look at some of my papers, that is how I think that the preventative vaccines for tumor-associated antigens are actually going to work by targeting molecules that are used by natural immunosurveillance of health to correct to prevent tumor from progressing. This is how the immune system does it. I don't like, people use terminology like reverse immunology or something. We'll tell the immune system what it should see. I don't like that at all. I want to see what the immune system has seen and then just make it see it again. So we can learn a lot from looking at what are the targets of natural immunosurveillance. And I think that these people, like you said, we identify people who have these targets. What we're doing now with the samples from this trial, we're looking at their repertoires, B cell and T cell repertoires, because the hypothesis would say that people who are responders to MUC1 may also, and then those who are responders naturally, may have enhanced the repertoires because they're all memory, they're lifelong exposures, and have larger repertoires that they can handle a disease and can also respond better to a vaccine. So you're asking a very, very precise question.
Thank you very much. Very nice talk.
Thank you, Bruno.
We have more students' questions in the chat. Lucas is asking, in the same vein of the previous question about adjuvant therapy, I wondered if you considered applying anti-inflammatory drugs maybe prior to the vaccine dose and or currently in the most advanced stages.
Yes, I think one of the thoughts was to, NSAIDs, right, non-steroidal anti-inflammatories, especially in the polyp situation, have been shown to reduce or delay polyp recurrences. So we were discussing that for sure. The problem with non-steroidal anti-inflammatories and steroids in general, right, is that their function is not limited just to like chronic, preventing chronic inflammation. It's actually, and they're not targeting the MDSCs very specifically, which seem to play a very important role here. It may not be in all. So we've been trying to understand how much we would then reduce also our general response, because you still need to present the antigen through a dendritic cell, that's activated, et cetera. So I'm more worried about immune suppressors than some other, more specific agents. But it would be, people have suggested we do aspirin, just aspirin, and we might consider it,
Also, Gabriela Maciel asks if the solicitation of an immune response with these antibodies could not cause a cross-reactivity against other proteins, relating this to the question of the difference in the response between patients also.
I'm sorry, let's see. She wants to know, so the antibodies don't react against normal tissues. In fact, I don't know if you have time, I'm holding you, but I could just show you a quick. So here, so we've taken, we've cloned out the antibodies from the responders. So here's one antibody, and it reacts very well with cancer, right? And this is a mouse anti-MUC1 antibody, so it shows where it should react. So it reacts to this, and here's isotype control. So they're very cancer reactive, and they also not at all against normal. So here's a mouse antibody that sees normal MAC1, low-level expression, normal breast, normal pancreas. You see this here, but our antibodies don't see it at all. So it's against MAC1, but it's against the abnormal form of MAC1. So we've actually published on these cloned antibodies. So it's very interesting when you immunize a person, even to a tumor-associated antigen that's expressed on normal cells, the person will not react, will not make a new antibody or something that is cross-reactive to, so autoimmunity is not dangerous because it doesn't happen. People don't respond with an autoimmune repertoire. And I can't remember the second half of the question, I'm sorry.
I think if this could be related to the responders and non-responders, or something like that, as I understand.
Yeah, I see what you mean. You mean that the non-responders didn't want to generate the autoimmune response. No, I think that so we've done a lot of work with responders, non-responders, single cell RNA, all kinds of things. We're actually preparing 2 papers for publication. I wanted to focus on biology rather than all the RNA-seq and single cell RNA-seq that tells you a lot and nothing at the same time. But the only thing that we can biologically relate to the response, age did not, females responded better than males, that's for sure. And this is an interesting fact in its own. The only thing we could relate was immunosuppressive microenvironment.
Gustavo, do you want to ask a question? Oh, Miriam, you can go.
No, it's very quick. It's just on that same line. Did you see a HLA bias?
No. So in the 100-mer, so we typed all our patients. In the 100-mer sequence, even though it's a 20 amino acid repeat, there are so many combinations because of the joining of the repeats that we basically typed for HLA class 1 primarily, typed the class 1 of the responders and non-responders and placebos, and we couldn't see any combination that worked. That was, I was kind of hoping that would be the case, and then we can narrow down, who is a responder, but that wasn't the case.
Gustavo?
I have a bunch of big questions, but I'm gonna start one more general. I really like the idea of the innate immunity is taking care of the first stage of the transformation of certain cells. And I also really like your macrophages being the surveillance, making part of this immune surveillance. But also you said that, the most probably what happened is like an equilibrium, much more than the elimination of this. And then combine this idea, combined with the fact that you're suggesting that the adaptive immune system is already in place as well. So your vaccine, for instance, is just, waking up. Have you tried to, or think about what would make this innate immune system to become useless in time and then make these cancer cells grow? And whether like something very simple as interferon gamma produced by, the adaptive immunity strengthen the innate immunity?
Yeah. Yeah, Gustavo, I think all of these things happen and people have documented them actually, so, but if you look at, for example, what is my friend? I'm forgetting my friend's name, Mantovani, Alberto Mantovani, who is, done most of the work with macrophages, right? Part of his work has been in the various mutations in the tumors that create now a pro-inflammatory tumor cell, right? So that you know, the macrophages may want to do what they want to do, but what the tumors make keeps them, you know, creates tumor-associated macrophages from them. So, so I think that, you know, that is that interplay, the macrophages and the tumor cells. The tumor cells, unfortunately, macrophages are more stable. They don't mutate, right? The tumor cells, unfortunately, constantly mutate. And so in those mutations, you'll get a tumor cell that starts to, you know, avoid all this, avoid recognition, or I don't think it's avoiding recognition because the molecules we're identifying are like, you know, housekeeping molecules, right? And, but certainly many other things, and, you know, the tumor metabolism is, you know, goes nuts. I mean, all the lactic acids, I think Miriam who mentioned it. So I think this is that selection. I think this is what happens. So the idea is to not allow the tumor to do that, you know, to nip it in the bud. I mean, as soon as it starts, get rid of it, because you let the tumor progress, it'll eventually escape.
So you don't believe that the difference, for instance, the non-responder to responder would be exactly the fact that some of these people, some of us would not have a anti-tumor response and others have. So you're waking up in those, the responders, but the non-responders doesn't have from the beginning.
Yeah. So I think it's a bell-shaped curve.
Right.
So I think we all have it. Because we all had, you know, chicken pox and mumps, and we, you know, the kids don't get them now, but we all had, it requires sort of very strong febrile infections to really, you know, fix in the immune memory. So we all had different levels of it. And I think you reach adulthood, again, with different levels of it. A lot of us are, you know, there, but some people very low, right? And some people very high. So the very low ones are probably the ones that are most susceptible to, if the tumor starts to grow, to tumor progression.
Okay.
And have a higher risk. So, yeah, there are now lots of collections of samples that have been collected, you know, from people at risk and, you know, until and after they develop cancer. So we have a chance now to look, you know, at immune biomarkers of, you know, of who is going to actually, whose risk is going to be a risk and who is going to overcome that risk. You know, lots of smokers who don't get lung cancer, right? And so, so we want to look at that. There's now our ability to look at it. This has gotten to be better. So we'll see what happens.
Just a quick question to finish, because you mentioned also infection. Some people are, you know, suggesting the use of attenuated pathogenic bacteria or virus at the tumor microenvironment.
Yes.
What do you think about that? And if this could fit in the awakening of
yes.
this?
So yes, so people are thinking of that in terms of generating, I mean, getting the pathogen-specific immunity to the site of the tumor, right? And in the presence of an ongoing immune response, you can awaken the tumor-specific immune response. I think it's a pretty good idea. I'm just worried that it'll be, you know, it'll work depending or not, depending on how immunosuppressive is the tumor microenvironment. You know, is it a hot tumor? Is it a cold tumor? Can you make a cold tumor into a hot tumor by doing that? Maybe, you know, so, but let's say you have a cold tumor. So there's no T cells in the tumor. You bring in the bugs and you bring in the bug specific T cells. You're going to have a hot tumor, but it's still not going to be tumor specific T cells, right? So I don't know how it's going to work. It's an idea worth trying, and I think it's very testable in animals. What I like though is if you, you know, what I would, if I had it, decision-making capacity here, I would make sure that every vaccine that we get, and our kids get, not we, but our grandchildren, and, you know, get, is an attenuated virus rather than a subunit vaccine. Because if you have an attenuated virus, attenuated bacterium, you know, BCG, nobody else gets BCG now, but it's a great vaccine, not against TB, but to prepare you for what abnormal cells look like at the site of the BCG injection, you know? And so you generate, it's those things that you generate. So I would prepare, and what we're hoping, you know, we're working on this very, you know, auspicious and very far goal that I probably won't be alive to reach, but for a universal vaccine, not based on a pathogen, not based on a tumor antigen, but based on these molecules that change in an abnormal cell. So every time something makes your cell abnormal, the immune system will get rid of it. But
sounds awesome.
It sounds awesome. So all these young people here who I don't see should do that.
Thank you.
Bruno, we see you now.
Yeah, and actually, I have another question, if I may.
Sure.
One thing that I find it very exciting to have this sort of prevention of cancer, but cancer cells are really nasty little buggers, and when we close an avenue, we may shut them down completely, but we also may open a window that they come out of it even stronger or nastier. So how did you deal with that? And I have another very specific question. What happened with MUC1 expression in the vaccine responders and those with adenoma recurrence? There is a change in MUC1 expression in these patients?
Right, so the MUC1 doesn't change. MUC1 is a very stable antigen. So those that didn't have the response, it isn't because the polyps were MUC1 negative. I mean, those that recurred, it isn't because they are MUC1 negative. Now, exactly what the reason that you brought up is I don't believe sort of in early detection of cancer. Okay, I say if what you detect early is cancer, it's not early, it's cancer, and it'll do what you said it will do. You can try to stop it, and unless there is a really large immune response already present in the person, that cancer will eventually find a way. You'll have a longer equilibrium, right? But you are a cancer patient. I mean, it's so I'm not crazy about early detection because I don't think it makes a difference. I think that early detection is just the first time you see the escape. You know, you see escape earlier, but it's an escape. So this is why, you know, you want to put on board a protection before it's a cancer that can evade the immune response. So it's pre-cancer. You have to do pre-cancer and eventually just risk.
Great, thank you.
You're welcome.
Miriam, before you go, can I just read the question from YouTube?
Yeah,
So,
I was going to read that, and then that was basically it. But and then another question came by. So when you finished it all,
okay.
I'll take.
So Professor Rafael Cardoso is asking from YouTube. Thank you for the brilliant talk. I was curious about the role of circulating MDSCs. Are there any antibodies to deplete at least part of these cells?
Not so far, because what they are is immature macrophages, immature neutrophils. So they are not different. They just have a different program. So one way that people are dealing with them is to mature them. So once they become macrophages, they're not immunosuppressive anymore. Once they become neutrophils, they're not immunosuppressive anymore. They're just blocked in their maturation. So this is why it's hard to get rid of them, but not mature macrophages, mature neutrophils, etc. So far as I know, it could be, there could be some companies already developing proprietary technologies, but in terms of the academic life, I don't think we have anything yet.
We have another student question in the chat. Fabian is asking, about the initial part, the focus was the macrophages and they are the THP1 cell lineage, and macrophages isolated from PBMCs. So in one moment, did you see the other effects of these PBMCs, for example, in other subsets as B cells?
So we only looked at macrophages. Yes, we only looked at macrophages. I think that so that the B cells will be second, right? So, you know, the macrophages will, and yeah, we really couldn't really do it. These were not complete cultures with T cells and B cells and macrophages. Yeah. Sorry.
Miriam?
Yes, thank you. So I was thinking about the preclinical use of the vaccine. So as you mentioned, the MOOC one, the changed one, the mutated one, is the one that is recognized. And I wonder, do you think there will be, is there too soon? Like if you mentioned, since you have people who have family history might be in line for that pre-cancer treatment. But do you think that if you do it too soon, the cells wouldn't have another stimulus and they would ended up fainting? Would you have to have a regimen of yearly vaccinating the patient, or would you require some sort of exam that would suggest that the person have started the process? Because as you mentioned, and with all this discussion, I agree completely that if you could get the system ready such that when the very first cells arise, that will be the best timing, right, to get rid of them.
Right.
But I wonder that if you try that, if you start too soon, would you lose them? And then the efficiency of the vaccination would be too variable, or
yeah.
How do you see that?
I have no answer to that, because it's just as complicated as the COVID. Look what we are thinking about, the regular, just a normal viral vaccine. You know, how long does it last? How, you know, I am a believer in lifelong memory. And I'm a believer in lifelong memory that is constantly boosted, right? So I'm not a believer in really lifelong memory. So some of them may be smallpox, you know, but look, you know, now they're saying that, you know, the people who have been vaccinated against smallpox are immune to monkeypox. So, you know, so there's a lot of memory. Now, I got a COVID vaccine and a booster, right? And I'm not getting any more. I don't believe I need a fourth and fifth and sixth boosters. Like, yeah, come on, you know, I'm not getting any more. But I'm also not constantly wearing a mask, because I believe, you know, I'm not going to kiss somebody with COVID, but I believe that having it in the environment in a low dose is a good booster. You know, we need to keep up our immune response by being exposed. I mean, we wore masks for 2 years, and now the colds that we are getting are the worst ever, right? And not COVID, just colds. So the immune response really is constantly challenged, and it needs to be challenged. And so I think some of these things that, you know, if with our preventative vaccine, we're boosting a pre-existing immune response. We are just bringing it out because there's a real danger now. There's something, you know, the immune response hasn't really handled it well yet. So we're just boosting it. But then I would think if a vaccine, if you boost and you boost in 1 year, which is what we do with a lot of things, then I think you would be protected. But look, you know, with tetanus, you want to do it every 10 years. So let's say you have a familial risk. So every 10 years, it wouldn't be a bad thing to get a vaccine, right? So. But
thank you.
It's all research, right?
And it's hard to test, I guess, right? Because
because for every question, it's a clinical trial, and the way it goes. So, yeah.
Thank you.
All right.
So I think we finished the questions for now. Gustavo, do you want to
ask one question? I want to say that was a wonderful talk. You know, I hope next time, very soon, we are able to bring you here to São Paulo. You haven't been to São Paulo.
São Paulo
is not as pretty as Rio de Janeiro, but we have a nice place to go as well. So hope that
wonderful.
We can make it possible soon. And thank you so much.
Wonderful. You're more than welcome. It was very nice to see everybody. Carla, it was nice to see you. And there may be some other friends I have but they're in the dark.
Yeah, the students are very shy. It's even when we do lectures for the graduate students, they like to have the camera close. You know, the excuse is always the connection is not so good, so we have to stay for the, you know. But it's, yeah, it's.
I think the COVID excuse was a good one. All right. Thank you all very much for inviting
Thank you,
me.
Professor Olivera.
Thank you so much.
Thank you very much.
If you see me at an international meeting or anything like that, stop me and tell me
Oh,
you were
yes.
on the top.
Of course.
All right, all right.
Bye.
Bye. Thank
See
you
you
so
next
much.
week.
Bye, everyone.